Hello and welcome to this session of ACS Science Talks, Connecting the World Through Science. This is the virtual lecture series, Scientific Talks by Specialists on Specialized Topics for a Specialized Audience. I'm Kunal Gupta and I'm pleased to be your host for today's broadcast of ACS Science Talks. Now, before we begin the session, it is my pleasure to invite Latrice Garrison, Chief Operating Officer, American Chemical Society, for an opening remark. Latrice, over to you.
Thank you, Kunal. Hello, everyone. I'm delighted to join you today to welcome you to our broadcast of ACS Science Talks. This is a platform by the American Chemical Society that allows us to connect the global science with global scientists. It's a confirmation of our commitment to advancing the scientific enterprise. As the Chief Operating Officer of the American Chemical Society, it is indeed my honor to interact with you today and to share our global outlook and mission to support science professionals around the world. At the American Chemical Society, we are dedicated to developing and providing the resources necessary for students, scientists, academicians, industry professionals, and anyone and everyone who's a part of our scientific community to thrive in this fast-changing world. Through our information products and services, we offer authoritative and indispensable knowledge-based solutions that empower science professionals to address the world's challenges with confidence. Our focus is on fostering innovation and effectiveness in scientific education. We provide access to opportunities, resources, skills, training, as well as networks to help ensure that scientists have the support they need to excel in their endeavors. By doing so, we contribute to the growth and success of the overall global scientific enterprise. It is our strong belief that diversity, equity, inclusion, and respect are fundamental values that drive progress. We are committed to promoting these principles as we work to identify and dismantle barriers to success. Our aim is to create a welcoming and supportive environment for all individuals, regardless of their background or circumstances. Together, we have the power to make a difference in the world. Through collaboration, knowledge sharing, and the application of scientific expertise, we can address the most pressing challenges facing humanity. By supporting science professionals like yourselves, we're laying the foundation for a brighter and more sustainable future. I would like to express my sincere gratitude to each of you for all that you do and for your dedication to the scientific enterprise. Your work is essential, and it's our privilege to stand together throughout this remarkable journey. I would also like to thank our guest speaker today, Professor Kelly Chibale. I heard his talk at the ABC Chem conference in Morocco last winter. He's an excellent speaker, an excellent scientist. I'm looking forward to hearing his engaging remarks today. Thank you to everyone, and many thanks to our ACS India team. We appreciate all that you do for the American Chemical Society. Thank you, Kunal.
Our speaker for today is Professor Kelly Chibale. Kelly Chibale is a renowned professor of organic chemistry at the University of Cape Town, UCT, South Africa. He completed his PhD at the University of Cambridge, specializing in synthetic organic chemistry under Stuart Warren's guidance. After postdoctoral experience at prestigious institutions such as the University of Liverpool, the Scripps Research Institute, and the University of California, San Francisco, in 2007, he was elected as a professor at the University of Cape Town. Professor Chibale's research group focuses on developing treatments for HIV, cancer, malaria, and hypertension, making significant contributions to the global health drug discovery. His remarkable achievements were recognized in 2018 when he was named one of Fortune magazine's top 50 world's greatest leaders. As an active ACS member since 1994, Professor Chibale demonstrates his dedication to our shared mission of advancing scientific knowledge and improving global health. Notably, Professor Chibale holds the distinction of being the first editor-in-chief of an ACS journal from Africa. He also serves as the founder and director of the UCT Holistic Drug Discovery and Development Center, or the H3D, the South Africa Research Chair in Drug Discovery at UCT, and the founding director of the South African Medical Research Council Drug Discovery and Development Research Unit at UCT's Institute of Infectious Disease and Molecular Medicine. Thank you, Professor Chibale, for joining us. The stage is all yours.
Thank you very much, Kuna, for that very kind introduction. I'm just going to share my presentation now, and then we'll get going. If you just give me a second. All right, can I just double check that my presentation is visible?
Yes, now it is visible.
Excellent, thank you very much. And Kuma, again, thank you so much for the very kind introduction and warm words. And thank you so much for this opportunity. The American Chemical Society appreciate the opportunity, and thank you so much, everyone who's tuned in to listen to this presentation. So over the next 30 minutes or so, I'm just going to walk you through a story around how we really targeting this very important superfamily or gene family of kinases in the malaria parasite for chemotherapy. But what I'll do is, before I get into my presentation, let me take the first few slides or so and tell you a little bit about our drug discovery center, which Dr. Gupta, Kuna Gupta mentioned, to give you a sense of the kinds of platforms that we have set up here at the University of Cape Town. So Kuna mentioned the H3D Holistic Drug Discovery and Development Center. So this is a center that we established back in 2010, based at the University of Cape Town and embedded within the Faculty of Science and the Faculty of Health Sciences at the medical school. So over the last 12 or so years, we've been on a mission really to do infectious disease drug discovery. And that's been the focus of our drug discovery center. We had to start the center from scratch. And so on the back of really executing drug discovery projects, it's really also building the critical infrastructure, which I'll come back to in a second. But secondly, we've also been on a mission to develop Africa-specific tools, preclinical tools, that really we hope when they're deployed will improve treatment outcomes in people of African heritage or descent. And as I'll show you in a couple of slides or so, we've also been developing drug discovery platform technologies that really are being utilized to have an impact on the various, phases of our drug discovery programs in all the projects we undertake at the Drug Discovery Center. Finally, but not least, we have been also on a mission to really train the next generation of African scientists who are skilled in integrated drug discovery, which is really about integrating chemistry in the broad sense with biology in the broad sense and pharmacology, including drug metabolism and pharmacokinetic studies. Last year, we had the honor and privilege of being selected by Johnson & Johnson as one of the 3 satellite centers that were set up across the globe. So we are a J&J satellite center for global health discovery with a specific focus on antimicrobial resistance, specifically precision antibiotics, focusing on developing first-in-class multi-drug resistant gram-negative therapeutics. So this is really precision antibiotics that we're after. So this is a significant development for our center, especially as you see on this next slide, that the company set up only 3 of these centers across the world. The first one was set up in 2021 in London, and that's focused on TB. And we were set up in Cape Town in April of last year, as I mentioned in the previous slide, with a specific focus on antimicrobial resistance, specifically gram-negative therapeutics. The third center was set up at the end of June last year in Singapore, and that one is focused on flaviviruses or emerging viral threats. I was referring to the technology platforms across chemistry, biology, and pharmacology, and these are summarized on this slide. So we have a medicinal, a chemistry platform, including synthetic chemistry, medicinal chemistry. We also have a computer-aided drug discovery platform, or CAD, to support our target-based projects, of which what I'm going to be sharing today is an example of how these computational tools are really becoming important in some of these programs. We've also integrated artificial intelligence and machine learning to build models using the data that we've been accumulating over the last decade or so across a variety of assays. And we've actually put a manuscript together that we hope we published in sometime soon. That's a chemistry platform. Then we have the pharmacology platform, which also includes the drug metabolism and pharmacokinetics platform, DMPK. So we have a suite of ADME, absorption, distribution, metabolism, excretion assays, a number of physical, chemical, and metabolism assays across the whole spectrum, as you would find in a pharmaceutical company. We also have, we do our own rodent pharmacokinetics to really understand the pharmacokinetic properties of a compound, which allows us, of course, to then design in vivo efficacy studies. Finally, but not least, underpinning all of this is the disease biology. Again, the areas described here, of course, reflect the therapeutic areas of focus. So we have malaria biology, both in vitro and in vivo. We have TB biology, and we have antimicrobial resistant screening. This is typically the WHO pathogens. There's bacterial pathogens that are in the priority list of the World Health Organization. We also have an enzymology platform where we do protein expression purification and setting up biochemical assays to support target-based drug discovery programs. And of course, we do cytotoxicity screening to make sure that the compounds that we're progressing, are really, we have a good safety margin and not really affecting mammalian cell lines. And of course, underpinning that is comprehensive data storage and sample management. All right, I'm going to shift gear now and focus on the main topic for today's presentation. And this is the subject of malaria, and I'll keep it very brief and very simple. So human malaria typically is caused by various species of the parasite we call Plasmodium. There are about 5 major ones, but the most virulent of these parasites is the Plasmodium falciparum species. The second one is Plasmodium vivax. So that's what causes human malaria. In any kind of drug discovery programs, the first question one asks oneself is, what is the unmet medical need? Why should we discover new medicines when we already have existing medicines available? When it comes to malaria, I can talk about the target product profiles in greater detail if there was an appropriate environment for that. But I want to focus on 2 main challenges for what we're looking for in the next medicines for malaria. Firstly, it's new drugs that are able to circumvent drug resistance. So drug resistance is really a major issue that really has the potential to render most of the drugs available useless, and malaria impossible to treat in many parts of the world. The second challenge is what we're looking for in new medicines for malaria are compounds that are able to kill the parasite at multiple stages. And I'll walk you through this diagram that you have very briefly to give you a sense of what I'm referring to by compounds with multi-stage activity. So if you look here, malaria, the vector for malaria is the female Anopheles mosquito. So during a blood meal, this mosquito injects what we call sporozoites. So these are parasites that are again released during a blood meal and spits out these parasites. They then get into the bloodstream and eventually end up in the liver here. When the parasites are in the liver, there are no clinical symptoms of the disease. So if we kill the parasites when they are in the liver, then of course we can prevent the infection from progressing to when the patient presents with clinical symptoms. From there, the malaria caused by Plasmodium vivax, which is often responsible for relapses. Some of the parasites go into dormancy. It's a bit like tuberculosis. 30% of the world's population carry the Mycobacterium tuberculosis, but that's dormant, and only as a consequence of risk factors that the parasites get activated that cause infection. So if we can find compounds that target these dormant forms of the parasites for the malaria caused by Plasmodium vivax, then of course we can combat relapse. Otherwise, from the liver, the parasites then begin to multiply, go into a form we call merozoites. So the ones that get injected into the liver are called sporozoites. They multiply, and then they begin to infect the host red blood cells. And it's really at the stage when the host red blood cells are attacked and infected that the clinical symptoms of the disease manifest, become evident. So therefore, if we can kill the parasites when they are in the host blood cells, then of course we can relieve clinical symptoms and therefore treat the disease. The other process that happens during the time when the parasite is in the host red blood cells, where it undergoes what we call asexual reproduction, is that there's another process that goes on, which is a sexual reproduction process involving what we call gametocytes. So these are basically sexual stage parasites. These are here is a male and a female. And essentially, this is what we refer to as the transmissible forms of the parasite. So during the next blood meal, when a female Anopheles mosquito, as shown here, feeds on a person infected with malaria parasites, it then takes up these gametocytes or transmissible forms of the parasites. And of course, through the mosquito, goes through a number of stages of development, and eventually malaria is transmitted from one person to another. So the key message on this slide is really that what we're looking for, in addition to compounds that are able to circumvent drug resistance, and typically those would be compounds that act by novel mechanisms or targets that the parasite has never seen before. Secondly, in choosing a molecular target for discovery, we're looking for targets that are expressed at multiple stages of the parasite life cycle, so that we can really come up with compounds that also can contribute not just to chemoprotection or relieving clinical symptoms, but also contribute to the eradication agenda by interrupting transmission of the parasite. So I've spent a lot of time on this slide, but this is really to just show you the context of what's coming up on the next slides. All right, this slide here really summarizes our entry point into kinase based drug discovery programs here, which I'll come back to in the subsequent slide. But the program really started with a phenotypic host cell screening of a commercial soft focus library. So a soft focus library is a library which is designed with a particular gene family. So this was about 35,000 small molecules representing multiple gene families, including G protein coupled receptors, ion channels, and kinases, et cetera, And one of the libraries that we prosecuted was what we called a soft focus kinase library. And what you see, it contained compounds belonging to this aminopyridine scaffold that we really progressed through the hit to lead optimization, and eventually selecting a candidate, which is this compound here, after making about more than 400 different derivatives. Came up with this compound that progressed to phase 1 clinical trials and then progressed to phase 2 clinical trials. But what we did in parallel, because we didn't know the mode of action or the target of this compound, so we really embarked on mode of action deconvolution, using a combination of genomics by generating resistant mutants, which we then sequence using whole genome sequencing, and then identifying genes where mutations occurred. And then we also took another complementary approach using chemoproteomics to basically pull down the targets of this compound here. Both the resistance selection approach, the genomics approach, and the chemoproteomics pull down approach identified a kinase we call Plasmodium phosphoinositol 4 kinase, or PI4K, which is a lipid kinase as the target of this compound here. And based on the knowledge of the target that we deconvoluted from these studies, we were able to develop a backup compound in about 2016 as a preclinical candidate here. Eventually, this really provided us with multiple opportunities to explore the entire malaria kinome for novel targets because of the implications of kinases in the life cycle of the malaria parasite, which is what I'm going to summarize on the next slide. But before I get there, just to show you very high level is just appreciating just the major difference between human kinases and malaria kinases. So the human kinome is huge. So we have more than 500 human kinases, whereas the malaria kinome is quite small. About 90 or so malaria parasite kinases have been identified. And then to the right of this slide, you will see quite a few phosphoinositol lipid kinases, which represent the family to which PI4K belongs. So we have PI4K, 3 different types of PI4K, we have PI3K, et cetera, et cetera. And so this is actually well covered in a review, a perspective article we published a couple of years ago. But the evidence from genetic studies so far suggests that 30% of the malaria parasite kinome is essential for the development of the malaria parasites when they're in the host red blood cells. It really gives you an idea that it's a much smaller family compared to the human kinases. But why are these malaria parasite kinases attractive as drug targets? Firstly, clearly from the work done in the oncology field, kinases in general are readily druggable. But in the context of the genetic work that's been done within the malaria parasite and the biology has been really elucidated over the last decade or so, a number of these kinases are essential to the survival of the malaria parasite. But also in the context of the malaria elimination agenda, to identify compounds that kill the parasite at multiple stages, is that many of these kinases are expressed at multiple stages of the parasite life cycle. That makes them really attractive in terms of really coming up with compounds that have multi-stage anti-malaria activity. The other advantages are really here, that they provide an opportunity to exploit polypharmacology, which really has numerous advantages if you can achieve it in a very selective way. And this is an example I'm going to exemplify before I finish off my lecture today. But also, given many of these protein structures that have been solved, especially the oncology field, that we can really exploit the protein structures that are available to design small molecules that are selective for the human malaria parasite and not really harming or going after the human ortholog. And there are several kinase focused libraries that are available for screening that we can screen to identify new starting points for malaria drug discovery. And also something we've been doing much more recently, which is not the subject of my talk today, is really this superfamily of enzymes really presents an opportunity for targeted covalent inhibition. And as I showed you when I gave you that historical context of how we got into this area, PI4K, the target of the compound I described earlier, proceeded to phase 2 human trials. Therefore, we have a very high level of validation of this particular target that really provides impetus for pursuing kinase targets in malaria. But of course, when you're talking about kinases, people often think they are a liability. And the main issue of concern is selectivity over the human ortholog. That's obviously has to be recognized upfront. But the second challenge, of course, where there are challenges, there are opportunities, is the risk of developing resistance as a consequence of mutations in the binding site of the kinase, which of course is something that one has to keep an eye on as you progress. And the final challenge, but also an opportunity, is that many Plasmodium kinases are yet to be studied and validated to varying degrees, which of course is the opportunity that's available for people in biology and drug discovery. All right, so we are taking 3 complementary approaches to targeting Plasmodium kinases for malaria drug discovery. The first approach is really screening kinase libraries that are out there so that we can identify chemistry starting points for drug discovery and then do mode of action deconvolution to identify the kinase targets. So it's identifying the chemotype and kinase target pair. The second objective of our program, which is the focus of today's lecture, is understanding the role of polypharmacology. What is polypharmacology? It's really identifying a small molecule that modulates or goes after more than one target. So it's really identifying multiple kinase targets that we can hit with a single compound. And as a consequence, we can really look at which of these targets really provides synergy or an additive effect, which also really allows us the opportunity to explore what are the other contributing mode of actions for these kinases. And as I mentioned in the previous slide, we're also involved in another program on targeted covalent inhibitors for Plasmodium falciparum. In the remaining time, I'm just going to focus on showing you a couple of examples of how we are trying to understand polypharmacology, identifying multiple kinase targets, and also looking at small molecules that not only go after a kinase target, but also act through other mechanisms of action, example that I've given here, impacting the hemoglobin degradation pathway. All right, the first example is a program that we did in collaboration with GlaxoSmithKline, GSK. And the specific aspect of this program was to study the effects of Plasmodium kinase inhibitors in the stage of the parasites when they have infected the host red blood cells. And then in parallel, do mode of action deconvolution, but also not just undertaking mechanism of action studies, but also undertaking mechanism of resistance studies, so that we can really begin to understand what is the potential for the compounds to generate resistance. And of course, that allows us to predict the resistant risk and help us to prioritize compounds that have a low propensity to generate resistance. So the background to this work that we did with GlaxoSmithKline is that the company screened 84 non-human kinase inhibitors using the so-called kinobid technology. What are kinobids? These are basically matrices which allows a human kinase to be immobilized onto a bead. And this technology has been used to study more than 500 human kinases. It allows you to study the interactions between a small molecule and a human kinase. And this technology has been used to capture so far about 54 or so of malaria parasite kinases. So what GSK did through Cellzome was, first of all, screen the 84 human kinase inhibitors. These are cancer drugs that are in clinical development, at least phase 2 clinical trials and upwards. Test those human kinase inhibitors for cancer. Test them on the malaria parasite to see which ones kill the parasites. And then in parallel, do mode of action deconvolution to identify the targets of those compounds. Out of the 84 oncology drugs, kinase inhibitor, anti-cancer kinase inhibitors, 16 compounds had the targets identified successfully. And so what we then progressed to do was to do extensive host cell studies on the parasite, using various tools, including looking at how the compounds interact with the different stages of the parasite life cycle. We look at whether there is cross-resistance with existing mechanisms. And the second objective we do as part of not only deconvoluting the mode of action or mechanism of action is to do in vitro resistance selection so that we can identify genes where mutations are. And so not only understand the mechanism of resistance, but also the mechanism of action. And the third component of this, in the context of looking at other contributing mechanisms, is to look at what is the impact of these kinase inhibitors on the host hemoglobin degradation pathway. And that's what we do with the intracellular heme fractionation, which I'll come back to on a single slide. And then we look at cellular accumulation to look at the extent of how these compounds are accumulating within the parasite and see whether that can be correlated to inhibition of the targets that are involved in mechanism of action. All right, one of the compounds that came out of the screening by GlaxoSmithKline of the 84 anti-cancer human kinase inhibitors was this sapanisertib, which is an mTOR, mammalian target of rapamycin inhibitor. This is an atypical phosphor, it's a lipid kinase, it's a phosphoinositol 3 kinase protein, and it's a key regulator of a range of cellular processes. But there's no clear human ortholog or no clear ortholog in the malaria parasite. So this is a compound that was identified, and some of the data I can show you on this slide here. So what we did was test the compound on, this is a resistant strain of the malaria parasite. Remember, one of the objectives of the drug discovery programs here is to really find compounds that are likely to circumvent drug resistance. So we tested on this strain of the malaria parasite, which is resistant to many of the drugs that are used clinically. And we found that this had quite good potency, about 70 nanomolar I50 value. We also tested this compound in the humanized mouse model of malaria. This gave us an excellent ED90 value of less than 1 milligram per kilogram. And we also could demonstrate that this compound showed activity across multiple stages of the parasite life cycle. And I'll come back to this and show you on a different slide. But we then also undertook mode of action and target identification studies, as well as mechanism of resistance studies to understand what we're dealing with here. This data that I've shown you on the right-hand side is actually data that GlaxoSmithKline, GSK, generated using the Kinobeads technology. And you can see that there are several kinases that were put down using this technology. And more than just kinase targets, there were also other proteins that were implicated in, that were binding to this compound within the malaria parasites. So our job was to really validate which of these kinases were really driving the antimalarial activity on the malaria parasite. And what we found was that, in fact, the major drivers of the Plasmodium activity, by correlating the inhibition of the enzyme to the host cell potency, the antiparasitic activity on the parasite, we found that the major drivers of the antimalarial activity was these three proteins here. So this one is the PI4K, which is the target I referred to at the beginning. This is also a lipid kinase. And you can see this particular compound is very potent against PI4K at 4 nanomolar. But the other contributor, big contributor to the antimalarial activity, is this protein kinase G, which you can see is also very potent. And also the other lipid kinase, PI3K, also contributing immensely to the antimalarial potency. And so this was verified, and we could show how this, in the left-hand side here, you can see how this compound here really interacts in this homology model. You can see this hinge binding with this part of the molecule here, very characteristic of ATP binding kinase inhibitors. And on the right-hand side, we can show so very clearly how it interacts with the, this is actually an actual structure of the PKG. You can see, again, there's your hinge binding interactions here in the ATP binding pocket, and there's interactions with the catalytic lysine that you see with this amino group of the benzoxazole moiety of the compound here. And so this data really verified that these two lipid kinases and this protein kinase were really big contributors to the Plasmodium activity of this compound here. And we've taken this program forward to design compounds that selectively inhibit at least two of these targets, PI4K and PKG, as part of the polypharmacology that we are really exploiting to maximize the potential to find compounds that are able to really have an impact on malaria chemotherapy. So this is just one example of exploiting polypharmacology, where we identify small molecules that go after more than one kinase target. The second and final example, before I give that example again, just to illustrate, by the way, that we could show also that this compound really had activity across the entire life cycle of the malaria parasite. We could see that it kills the parasite at the liver stage. Therefore, this compound potentially could offer protection. And of course, that it was able to kill the parasites when they are in the host red blood cells. And I showed you this data already. But also they're able to kill these transmissible forms of the parasites, particularly the late stage gametocytes, which are the ones that are really implicated in the transmission of the parasite. And we could show further that this compound was able to really inhibit the development of the parasite within the mosquito. So really excellent profile, not just in terms of killing the parasite at the different stages of development within the human host, but also killing the parasite during its development in the vector, the mosquito. And that's data that we generated with our colleagues at UC San Diego, but also here in South Africa, in Johannesburg and Pretoria. All right, the final and second example I wanted to share on the polypharmacology is compounds that inhibit a kinase and other contributing mechanisms. Many kinase inhibitors that are described today bind to the ATP binding site of the enzyme. And in fact, many, when we generate resistant parasites, many of the mutations that we see in many kinases, really mutations are in the ATP binding site. But when we look at many of the kinase inhibitors, what you see structurally as a chemist is that many of them are characterized by these structural features. They tend to have heteroaromatic ring systems. They tend to be planar structures, and they tend to be weak base compounds. You can just look at many of these structures, and if you compare them to this famous drug used in malaria treatment, chloroquine, it's also heteroaromatic, and it's got these two basic centers here. So that's a similarity. If you compare that to the way chloroquine works, to keep things simple, chloroquine really works by interfering with the host hemoglobin degradation pathway. So this is your host red blood cell here. And this diagram is simply showing the parasite inside of the host red blood cells. And it begins to break down the host hemoglobin in order to derive amino acids for protein synthesis and replication. But one of the byproducts of the hemoglobin degradation pathway is this molecule here, heme. Heme, here the ion is in the +2 oxidation state. And that undergoes autooxidation to hematine, in which the ion is in the +3 oxidation state. So the way chloroquine kills the parasite is by binding to hematine and therefore preventing the conversion of hematine into hemozoin. So heme and hematine are toxic to the parasite. And the parasite has a natural way of detoxifying this material. But when you block the conversion of heme or hematine to hemozoin, then of course the toxic hematine and heme builds up and kills the parasite. If you compare then some of the non-kinase inhibitors that are described in the literature for malaria and oncology, you can see that a number of these kinase inhibitors also really have been shown by our colleagues here in South Africa that there's an overlap, for example, between a number of these PKG inhibitors that are described in the literature and kinase inhibitors and those compounds that interfere with the hemoglobin degradation pathway. So this really gave us an opportunity to explore some of the compounds of the GSK family to see whether we can show polypharmacology. And this is summarized in this particular slide here. It's a busy slide, but the message is very simple. On the left-hand side, I made reference to the 16 kinase inhibitors and the various malaria parasite kinases that we identified. These are summarized here. It's quite a long list of kinases that were identified from the work we did with GSK. We then had to validate those targets here because these were basically binding assays. We had to really put these compounds through biochemical assays to provide an additional level of validation. And we saw that we could really identify a number of malaria parasite kinases that were implicated in inhibition of, in the antiparasitic activity here. We also could show, using conditional knockdown lines, that we could validate that these compounds were really on target. And this is the work that we did with Jacqueline Niles at MIT. So what we've done, which is not something I'm describing today, is that this work here is now formed the basis for medicinal chemistry optimization that is ongoing, but also has formed the basis for the targeted covalent inhibitor approach that I described earlier on. But the final point here is that we could demonstrate, and I'm not showing structures on this slide for confidentiality purposes, but what this slide is demonstrating here in very simple terms is that if a compound really interferes with the hemoglobin degradation pathway, you expect two things. You need to see an increase in the concentration of free heme. So the concentration of free heme is what you see here. With increasing concentrations of the kinase inhibitor, we can see increased levels of the intracellular heme, which of course is a feature of compounds that bind to heme and prevent the formation of hemozoin. The second feature that you see of bona fide hemozoin inhibitors that you should be able to see a decrease in the intracellular levels of hemozoin. So this was a powerful validation that these kinase inhibitors that I described earlier are also acting by interfering with the hemoglobin degradation pathway by preventing the formation of hemozoin. This is my final slide, and I really just want to acknowledge the various collaborators. You can see this is a huge team of collaborators from H3D, from the University of Pretoria, and all the institutions listed here have really been big contributors to this program. And of course, special thanks to the Malaria Drug Accelerator. This is a consortium that is supported by the Bill and Melinda Gates Foundation that has multiple groups and industries working together in malaria drug discovery. Thank you so much for your attention.